what we are talking about, that we stopped at this slide, that the viruses hijack the cellular machinery to replicate and reproduce. And the difference is that the first thing that what we are seeing, the top panel that on the right-hand side, what you are seeing, that this is a typical growth curve of a bacteria. And most of you have taken the requirement, which is the micro 3050. So you have studied this growth curve. And again, this is not complete. You know that it becomes flat and then it just comes down as well. So it is not complete, it's just a part. But what is important is that bacteria, living organism, initially when you have a media, and in that media, which is a nutrient media, you add your bacteria, you see a kind of a flattened growth initially. And this part is called the lag phase, which is basically when the bacteria is adapting to the environment, just adjusting itself, just what they need to do, these kind of things. After that, you see that there is a log phase and there is an exponential growth just like this, 1 to 2, 2 to 4, 4 to 8, like that. So exponential growth. And then what happens that gradually it plateaus out, which we call as a stationary phase, when there are two things, that the nutrients are depleting, there is not enough nutrients, and also there may be accumulation of toxic waste products. And the plateau means that the number of cells being reproduced is equal to the number of cells dead. This is how it works. So that's a typical bacterial growth curve. But the virus reproduction, and again, remember that we are just using the anthropomorphic term, virus reproduction or the replication do not follow that pattern, because they are not like binary fission-like cell. The way they work, that after infecting the cell, they make all the different components, and then finally they assemble all it together in a concerted manner. And then they will either make the cell lyse, burst. Some of the viruses also bud out of the cell. So when you infect a cell and just look at the viruses that is present in the cell after the infection, you just don't see the virus. So you infect the virus, the cells, and you see that in the cell, if you just measure, so you just take many samples and you just take time points. At a certain time, you take the cells and try to break open the cells and measure the viruses. And as you are doing that, you see that suddenly these viruses have disappeared. You just only have just a little bit of, see that it is just not at zero, because you have this much of virus that you have added in there. So that's why it is not right like there. And they have disappeared, the viruses have disappeared from the cells. Just only the amount of virus that is available that you just infected with this. And we call this as an eclipse period. And in the eclipse period, they are doing a lot of different things, and that is we are going to dissect out all throughout the course, that what they are doing at that time. And then suddenly what happens, so you can see an extended amount of eclipse phase, just not a small lag phase like this. And then suddenly you see just an exponential growth, which we call as a burst. So these viruses just come out as suddenly appear, all of them, and in an enormous amount, or constantly. If it's a cell, it will lyse the cell, come out, and or it will just bud out of the cell constantly. But again, you will see a burst of virus. So that's the basic difference in between a living organism growth curve, just like in bacteria, and a virus. So you can easily see that it is not a binary fission that you're seeing right here, whereas and this is only possible because the viruses make all the parts and assemble the final product. When the final product is assembled, they just all come out together. Only at that time, you detect the virus. So you can see that just from one virus, that before that, you cannot see an infectious virus particle. You will see that some of the parts of the viruses are made, but they are not infectious virus particles. So this is how the basic machinery, the viral machinery work with the hijack the cellular machinery to replicate or reproduce. So a couple of other interesting things is how old are the viruses? So you can easily see right here that there are some Orthoceras, some nautiloid cephalopods, 488 million years old. And it is just suggested that some of the retroviruses are probably 450 million years old. And how do we know this? And again, remember that we said that these retroviruses infected all these different animals, and they have become a molecular clock inside their genome. So we can just get some of these DNAs from the fossils and those sediments, and we can analyze those, and we can just find some of these sequences over the period of time. That is one of the way. On the other hand, we can see that some of the more recent species, and look at their genome sequence, and they also have the retroviral sequences over the evolutionary process. And there is an algorithm, very complex, that can also shed some light that how old they are, how they're related. And by a complicated process, you can just back calculate these numbers that when they are. So approximately in the Ordovician period, it is understood that at that time probably is the latest that we can go to see some of these viruses or the retroviruses. Now, the thing that we don't know is that whether they came before the cell or after the cell. We cannot answer that questions. The reason is that, as I said from the very first class, that they're obligate intracellular parasite. If they're outside the cell, 450 million years ago, that small chemical particle is almost impossible to have that one. You cannot get a viral fossil as such. Probably you can, but you just cannot detect it. It is just not possible to do that. So you have to have a cellular entity. And here we are having some of these animals which are fossilized or partially fossilized. Still, you can get some material from them. That is a possibility from where we can just derive. So you have to have a cellular entity. So we can just only go back after the cells have appeared, but the viruses probably appeared before that, and they have a chemical entity. So the many different chemicals came together and form a molecule. They were just like a passive agent, like a grain of sand sitting at the bottom of the ocean or land, and did not infect anything because they just do not have any means to replicate. As the cells evolved, somehow they gained access to these cells and just could enter the cells to replicate themselves. That's a possibility. That these are all speculations. So some of the ancient references to the viral disease, and there was some this is from the Iliad, and you can see that rabid Hector. There was in the bottom in the notes, it was written as a vector. So there are lots of these kind of typos in there. So I just corrected all of these. Rabid Hector, that's one of the references. So the rabies were noted as a disease, but again, remember that these are viral disease and a reference of the disease, but no information about the agent as a virus was known at that time. No concept of viruses. And here you can see that from the 1580, 1350 before Christ, this is engraved from Egypt. And if you look at the leg of this human being, a priest, you can see there's some deformity in the leg, and most likely that this is poliomyelitis probably, and this is probably polio. We think that that's what it is, and this is some of the ancient references of that, but really no idea of the disease. And then the immunization. And remember that we will have a whole chapter on vaccine. We're going to talk about details about that. This is just an introduction. That in China, in the 11th century, variolation was a process. So what the people used to do, that if they have smallpox that ravaged the whole world, they will take a needle and then puncture a pustule, take a little bit of pus from that, and then put it somebody's arm on the skin. Unfortunately, this is not a vaccine. Remember that this is not a vaccine. Many of these, as the process of variolation, will get full-blown smallpox and will die. So that was a sad part. But if they didn't get the severe disease, they got immune to it. So this was practice. And then this lady Montagu, she was the wife of a British ambassador to Turkey. At that time, the Ottoman Empire, they were also doing some kind of variolation. So there are some differences in there. Sometimes what they will do, that they will just scratch some of the skins next to the pustule and inject those different ways, and not going into the detail of that. But that was the idea. And Lady Montagu came back to England and just told people to do this idea. And at that point, people started doing this. But again, that was extremely dangerous. A lot of people actually got smallpox, and due to that, full-blown smallpox due to the variolation, and that was not a perfect disease. And then 1790s, ultimately, the Edward Jenner basically established the vaccine, and we're going to learn about that whole story in the vaccine chapter. And many of you probably know that how it was done. So now, initial viral discovery. So tobacco is, of course, these plants were big economic impact all over the place. And people will see that sometimes they will have these blotchy tobacco plants, and they didn't like it. It just reduces the value of the crop, and this was a problem. So at that time, there was a scientist, her name is Adolf Meyer, at Wageningen University in Netherlands. It's an agriculture university, it still exists, very famous. Termed this as a tobacco mosaic, but he did not discover the virus. He just coined the term tobacco mosaic. And then gradually, scientists were trying to grind up the leaves and trying to understand what is caused. Many of the people initially thought, including Meyer, that it was probably caused by a bacteria. That was the best knowledge that they have. They have no idea about the virus yet. So after that, there was some more experiments done historically. And let's take a look at this. So in 1892, Ivanovsky is a Russian scientist. And these are some of the filters which we called as a candle filter. And these were made of porcelain. And they have the 3 grades of porosities. There are many different names. This is a Berkfield. There was Chamberlain. You'll see that many different types of filter. And these, depending upon the pore size, there are 2 of these grades of these porcelain filter, which will keep the bacteria. There was no bacteria can go through them. So now we know that you have to have a pore size of 0.2 micron. So remember that value is 0.2 micrometer. That's pretty much, if you have a pore size like that, it will contain any bacteria. So what he found, that the filtrate, which is coming right here, contains an agent which is still infectious. And then 1898, Bajorink, following his experiment, actually named this as a Contagium vivum fluidum. And what they did, that they crushed up these tobacco leaves, just passed it through these candle filters, porcelain filters, took the filtrate, and then he found that in that filtrate, that agent doesn't replicate or do anything. But if that is inoculated on a tobacco plant, they get the disease. And that was the start, basically, and they just named this as the virus slimy liquid poison. And you can see that these are the pictures of some of the modern tissue culture, cell culture. If you just work in a laboratory, you're going to see that. So basically what you have, that these are some of the media with an indicator, pH indicator. Just right here in the middle, in between the top portion is where you pour the sample. And then right here at the intersection of that you have the 0.2 micron filter. They are made of many different things like nitrocellulose, polyvinyl pyridine, many different material. 0.2 micron filter. And then this is a vacuum manifold. So you have to vacuum, put them under the vacuum. Otherwise, no liquid material will pass. The pore size is too small. You have to suck it out through that, so doing a vacuum. So the same thing right here. And the liquid will go through that. And here, you can collect the sample. Now, this is routinely done in the laboratory, and this will get rid of all the bacteria, which is fine. But if your media is contaminated with a virus, it's not going to do anything. So most of the time when you're using a clean room, that is not the case, but that's what it is. In the modern days, we use these bacteriological filters as well. All right, so some historical things. 1898, the agent of foot and mouth disease was found. These were the filtrable. And the key concept that came out that that's not only small, but they replicate only in the host, not in the broth. And still we just know this, that it is that's what they do, that they are obligate intracellular parasite. Host means that the host have living cells, which can be plant, which can be a bacteria, which can be a protozoa or an animal, larger animal or eukaryote, but they can just only replicate in there. All right, so these are some of the historic perspective. And again, remember that in this class, we will ask some of the questions, as I said, just a few questions where you just remember, understand, as I said, but not like this. I will not ask you a year when it was discovered, but 1901, at this time, the Panama Canal was being dug at that time. And yellow fever actually broke out, and that was the discovery of the yellow fever. And you can see that many of the viruses eventually were discovered in that. And we're going to see that in 1911, this Rous sarcoma virus was discovered in 1911. And we will see that from that virus, many significant discoveries were made from that Rous sarcoma virus, which causes a tumor. We're going to talk about many times. So the first oncogene or the cancer gene in the 1970s were discovered, which is called the SERC oncogene, was discovered. That was the first oncogene, that a gene responsible for cancer from that virus. That was the first discovery. But the virus was discovered in 1911. The gene was discovered in 1974. So you can just well imagine the implication of research sometimes takes decades in there. So that's pretty self-explanatory, some of the milestone of the virus. And these are also some of the era of virology, started more than a century ago. You can see that gradually the birth of a virology, pathogen discovery, then plant and chemical period, early years of bacteriophages, which infect the bacteria. And the phage, basically the bacteria, opened up the process of our understanding of DNA replication, transcription, translation, as well as mRNA splicing and other. And the birth of molecular biology was triggered in the 1970s due to this, because you can label a virus and track it, and you can tell that what exactly it is doing. So that was the idea. And then 1950s, 70s enhanced our knowledge and the concept of virology tremendously. So here you can see that the discovery trend of the microorganism, you can see that almost all of them does have a gradual growth. And then you can see that right here, 1875 onwards, the bacteria discovery went through the roof. But right here, you can see around 1930s, the discovery of the electron microscope basically caused the viral discovery to go on. And the first time with the electron microscope, people were able to see the viruses. So here, just a couple of pictures of the electron microscope. So a bacteriophage, a tobacco mosaic virus. This could be an influenza virus, probably. There are many viruses look like this. So I'm just saying that influenza virus looks like this as well. So this is a bullet-shaped virus, very interesting. This is rabies virus, is exactly like this bullet-shaped virus. And again, I'm not going to ask you that which picture is what. And also another virus, which is a vesicular stomatitis virus, which is a prototype of the rabies virus that also looked like a bullet foot virus. I make my career or the postdoc research career on that. So I used to work with the vesicular stomatitis virus. At that time, I was a postdoc at the Cleveland Clinic Foundation. So we'll talk about those stories sometimes. All right, so here we are looking at some of the pictures, and we know many details, and we basically know a chemical formula of a poliovirus. You can easily see exact number of carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. We can tell because we know each and every protein on that nucleic acid, protein, nucleic acid, lipid, carbohydrate on that virus. So that is the detail that we exactly know. So what you are seeing in this picture is the background is a raw picture from a cryo-electron microscope. So the cryo-electron microscope basically captured these images, and they tilt the frame a little bit so that each of these virus particles are pictured from a slightly different angle. And then by the computational process, you can find out each and every alpha helices and a beta strands in there, and you can build your protein model in there. So as we are going through the years, the computational power went through the roof, and the cryo-electron microscopy is now replacing a process which is X-ray crystallography, where you first need to make the crystal, shoot it with an X-ray, and it will diffract that crystal, if it diffracts. So after that, you just get the diffraction pattern, and by which you call as we'll talk sometimes as a Patterson function, you can back calculate the electron density. And into that electron density, you can build the chains of the amino acids. So you will see that there's a ring in here, a side chain in there. Okay, all right, this is glycine. There's a big ring in here, that is phenylalanine. Here's a big two rings next to each other, this is histidine. You can build those in there. That was a classical method. But now but it was an extremely laborious process. Sometimes we will talk, and we used to call as almost a black magic, that X-ray crystallography. I did that for a long time, for 10 years. When I was after Cleveland Clinic Foundation, I was there for 3 years. Then I was there at the Case Western Reserve University. This is in Cleveland, Ohio, just next door, for another 7 years. And that time I did this structural biology work. And we'll talk about that in a separate chapter, some of these fascinating facts in there. So that's how you basically figure out the exact molecular structure of these viruses. All right, so the next part we are looking at is the classification. And these are some of the things, this is important for your exam as well, that on the basis of which you classify the viruses. And it is very difficult, or the classification is not as same as how you classify a living organism. It's very different. So the first is the nature and sequence of nucleic acid in the virion. This criteria doesn't even stand for any living organism, because the nature of the nucleic acid is always DNA. The genetic material is DNA everywhere. Here, the first time, you have DNA as well as RNA. Not only that, we're going to see that in the genetics chapter, there will be double-stranded RNA, double-stranded DNA, single-stranded DNA, then double-stranded RNA, and then plus-stranded RNA, which is just a 5' to 3', and a minus-strand RNA, which is 3' to 5'. You do not need that right now. We're going to talk about that structure. So many different types of nucleic acids. And we can see even a hybrid. Part of the genome is DNA and a tail with an RNA, incomplete genome. We even see that. After that, we are looking at, in the classification process, we look at the symmetry of the protein shell, which is known as the capsid. And again, we are going to define these terms in the structure chapter as such. So what is the symmetry of the protein shell? So if you look at this picture particularly, right here, there's a naked virus. And this one we call as an icosahedral symmetry. So these are the typical polyhedrons, and these polyhedrons is known as the icosahedral. They have 12 faces, 20 faces as such. You can just look at that. 20 faces, which are with equilateral triangle. That's one of the things. And right here, this is also an icosahedral symmetry, just a little bit bigger in there. And then right here, you have, oops, sorry. Right here, you are looking at an adenovirus, and that is also icosahedral symmetry. Right here, another virus, icosahedral symmetry. So that's basically, most of these naked viruses will have this. On the other hand, all of these enveloped viruses, if they are round or long or this kind of shape, all these enveloped viruses, we call this, they have a helical symmetry. We do not call them, they have a round symmetry, elongated symmetry is just one type, helical symmetry. That helical symmetry can represent a filamentous type, a round type, a complex type like this or this. So like a mumps virus, like Ebola virus, HIV, herpes virus, coronavirus, these are all with a helical symmetry. They do not fall under the icosahedral symmetry. And then presence and absence of a lipid membrane, which we call as an envelope that we talked about. So you can see right here, this virus, icosahedral, doesn't have any envelope. What you are seeing, this is made up of solid protein. Could be many proteins, multiple proteins. This is solid protein, naked virus. Here, the adenovirus right here, you can see that this is also a naked virus. It does have some, these are called the fibers for attachment to the receptors, but it is not enclosed in a membrane. This is naked. These are the naked viruses. On the other hand, all of them, what you are seeing, that has a round structure outside. See that inside there is an icosahedral shape, and that there is a round membrane around those. And these are the enveloped virus. So most of them, what you are seeing, surrounding that, most of them are with an envelope, presence of the membrane. And then also the dimension of the virion, as well as in the capsid, those are some of the classification terms as well. All right. So here we are looking at the virus classification system. Here is a picture like an original electron microscope and then a cartoon. This is a filamentous virus. This is a typical Ebola virus that you are seeing. And the classical kingdom is the kingdom, then we go to phylum, class, and the order usually ends with a term viralis. Say for an example, the Ebola virus, again, you do not have to remember it here. The Ebola virus have a minus sense, negative sense, minus sense RNA and one RNA. So it is the order is known as mono-nega viralis. Mono means single, nega means it's a negative sense RNA, mono-nega viralis. That's how it is. And then the family is Viridae, Filoviridae, in the filovirus family, filamentous virus. And the genus is the Ebola virus Zaire, and the species Ebola virus. Now, remember that one of the important thing that when you're looking at the influenza virus, that's the genus, and then you will say, okay, all right, we have an H1N1, H3N2, H5N1. What are these? Are these species? No, they are not species. Remember that they are not species. These classification are much, much below the species level. These are just the different isolates of the virus, and we can call them strains. But they are not species. So just make sure. So when we are looking at the SARS coronavirus as well, the genus stands with the alpha coronavirus, beta coronavirus, then delta coronavirus. The coronavirus that we have is a beta coronavirus. So that's where we stop. We do not go beyond that. Now you have this alpha variants, beta variants, delta variants, which are the many mutations. You have 12 mutations, and then you have the different ones isolated from like an AI1, AY2, 3. You can just look up that what those means. Those are the different isolates with about 10 mutation, but it goes by the code name B1.617.2. That's your delta. So all these numbers, what you see are way below species level. They do not represent the species. The species is still beta coronavirus. That's it. So here, if we just look at this ICTV online, this is just only for your little bit of fun right here. You can just take a look at this. So we are looking at. So if we're just looking at the taxonomy, hold on 1 second. I'm just trying to find it out right here. So here, I'm just trying to type that. You can just have fun with that. Say coronavirus, not saying. All right. So now you can easily see it's a ribovira, ortho RNA viri, then Pisuviricota, and this Nidoviralis. This is from the order. Then suborder, then you have the Coronaviridae family, Orthocoronaviridae, alpha coronavirus, then beta coronavirus. That's what it is. And these HKU10, 229E, these are your strains. So these are your strains below that level. So here you can see that if you just take a look at the overview of that, you will be able to see. So the thing is that this is the beta coronavirus right here. So you can see that all of the genus are the beta coronavirus, and there is really no species level in there. If you just look at that, there are 5 of these which are the different names and like a lineage of that. But again, remember that when we're talking about the specific ones, this alpha different, we call them as a variant, not a species. Just to have an overview of that, how the taxonomy basically works. So that's about it of the classification. So one of the important factor here is the virus discovery once driven only by the disease. We saw that 1901, why yellow fever virus was discovered at that time, because it was a problem. A lot of people got infected, and yellow fever used to break out in a long journey through the ships. Say, for an example, all these people from the Europe, when they were trying to migrate to the United States, sometimes yellow fever will break down, and they used to come to the Ellis Island in New York if you just go in there for their immigration check at that time. And if there is a yellow fever in a ship, they will just keep it in the sea and quarantine. And sometimes it happened, unfortunately, almost the whole population in the ship will be wiped out. So that was the discovery only by the disease. Now, the discovery of virus is entirely different. You just take an insect, crush it up, take the sample from there, or you just collect some water sample from somewhere, and you just send it for the next generation of sequencing. And this next generation of the sequencing, what is the advantage of that, which you call as a metagenomic analysis for doing that process, it can pick up, it is so powerful that it can even pick up just 1 or 2 copies of the DNA or RNA, what is in the sample. And earlier, this was not possible. You have to have a good amount of nucleic acid to sample that. And even if these DNAs are fragmented, the fragments are analyzed, and you have a computer algorithm to put it together, and you will just find out that what is a virus that is present in there. Many viruses were discovered this way, and you can easily see and take a look at the original thing that analyzed RNA from 220 invertebrate species, and 1445 new viruses actually were discovered. And you can just see the full Nature article in here. And this will, at that one point of time, will almost be saturated. And they're all, I mean, like, because people are just sampling every possible place and just trying to see that what viruses were there in these samples. Say, for an example, I'm just giving an idea that Caenorhabditis elegans or C. elegans, these are some of the nematode which are the model organism. And until 2015, there was no known viruses. And then in 2015, it was discovered that Caenorhabditis elegans can be infected by two different type of virus, is FHV, called flockhouse virus. So these discoveries are still going on. All right. So what is the reason that we really do care? So you can easily see that the viruses outnumber the cellular life by 10:1, gives you huge biodiversity, beneficial global biogeochemical cycles that we just saw, that there are 10⁻16 infections happening every second in the ocean. So basically what they do, that all these different bacteria, microbial life that you have in the ocean, the phages infect them and lyse the cells. And all these bacteria basically sink at the bottom of the ocean and recycle the nutrients. So that drives the biogeochemical cycle, and that's beneficial for the earth. And of course, this is not good news, but is a sources of new pathogen, and that's a concern. It says nothing like at the present time. You can just well imagine that how our lives have changed just by one pathogen. And the engineered viruses emerging as in nanomachines. Now, this single line, do not go into the conspiracy theories out of that. We can engineer a lot of different viruses. And these basically what they do, that for the experimental process, we can engineer a virus, we can just put a toxin into that virus, and we can target it to a tumor, because it will just specifically go to the tumor, deliver that toxin for the cancer therapy. So oncogenic virology is a process. There are several ones in the clinical trial, and this is a delivery machine. But again, just remember that engineering, we can engineer the virus, we can make a lot of recombinant, but a human pathogen, to engineer it for a specific way that it will become infectious more or something like that, you just cannot do it in the cell culture. Even with all this technology, we do not have the idea that it will. Say, for an example, we can see that over the period of time, just within this 1.5 years, the Delta variant, the virus evolved over the period of time to make that variant. And that will convince you that it's a natural selection process. So what happens, the virus is constantly mutating. Every replication cycle, everywhere it is mutating. And what happens that when you constrain the virus, it's not allowing you to escape, to infect new people, then only the ones which are more transmissible can go to another one, and that is the strain that will take over. And this is how the mutations are selected, just like the survival of the fittest. And this is how the viruses do. The humans of like those 10 mutations that is from the original SARS to that 10-12 mutations that you have in Delta variant, no human can engineer that. It's just some random. And the problem is that in the cell culture, remember that, this is very important. You have the cell culture as a mean, or maybe some animal models. The cell culture doesn't have any immune system. When the virus, so you need a human being to engineer a virus to see the effect of that. So I mean, like, there is no laboratory where you have a human behind the cage and you just infect them. Monkeys or chimpanzees, primates, their monkeypox virus has nothing to do with the human pox virus. They are different. Their susceptibility is different. So to really check out a virus, how infectious it is, you have to have an animal subject, like a human subject. I mean, not animal subject, human subject. A cell culture will not give you the exact definition, even if you have the animals, even if you take the lung cells, or say the epithelial cells from your nose, that you do it in a culture, you can propagate the virus, but what it will do in a human being, it doesn't translate. And that's the way things work. All right, so the last things that a take-home message that what you have is that there is an underlying simplicity and order to viruses because of the two simple facts. And this is your take-home message, the most important one from the first chapter, And that is that all viral genomes are obligate molecular parasites that can only function after they replicate in the cell. Other than that, it is a complete chemical entity. We talked about that. And all viruses must make the messenger RNA, can be translated by the host ribosomes. They are all parasites of the host protein synthesis machinery. So what is that turkey-like thing? The picture? What is that? Anyone? That is correct. That's a ribosome. The top one is the large subunit of the ribosome, and then you have a small subunit of the ribosome. The prokaryotic, eukaryotic, different ribosomes. But basically what it is, the virus has to make the mRNA. Once it makes the mRNA, that message is read by the ribosome of the host. The virus does not have still date. There are some viruses which still can make, some of these giant viruses still can make a transfer RNA or a tRNA. But there is not a single virus that we know of, because otherwise it will be. They cannot make ribosomal RNA. Our RNA cannot be made, and that's why they cannot make any ribosome or protein synthesis machinery. This protein synthesis machinery has to be in the cell, and that is what is hijacked. That's what it is. The only thing that has been done, that's only one example, that some of the scientists were able to replicate poliovirus in a broth or extract from a cell, but that extract has to have ribosomes in there as well, the ribosome and the other factors. It is a cell-free, means that it is not, that extract is not bound by a cell, and poliovirus can replicate in that, it has been shown, but it has to have the protein synthesis machinery in that extract. So we will stop here, and we will go into the next one. But before that, any questions?